Welcome back. This is Coach Heiko with another podcast over DNA. This time we're going to talk about DNA replication, why it's important, what happens, and where it actually takes place at. So let's get started. As a basic review from last time, Chargas Rule tells us that adenine is always paired up with thymine, and that guanine and cytosine are always going to be paired up together. And since they're paired up together, their amounts are going to be the same within any given DNA molecule. So we have our A and our T and our C to our G. So when we have questions like this, as long as we know how Chargas Rule works, it should be pretty easy to apply. If there's a question where we have 30% adenine and we want to know how much cytosine we have, well, this should be pretty easily accomplished. If we know that we have 30% A and we want to know how much C we're going to have, well, we're also going to have 30% T. Okay, so that's going to leave us with 60% A and T combination, each representing 30%. So we know we're going to have 20% cytosine, and this is why. So if we have 60% of the DNA molecule, which is made up of A's and T's, then we must have 40%, which is made up of C's and G's. Okay, so if we have adenine and thymine are both 30%, and guanine and cytosine are 40% combined, 20% apiece, we know that we're going to have a total of 20% cytosine. Make sense? Great. Let's move on. DNA replication is the process of making exact copies of DNA. And remember, this occurs in the S phase. If we remember that from mitosis, the S phase is the phase in which DNA was actually synthesized or made. It does this before the cell replicates because it wants to make sure each cell has the correct amount and the actual right DNA, so that there's no mutations or there's a less likely chance of mutations actually occurring. So replication begins with what's called strand separation, and an enzyme named helicase. We know this is an enzyme because it ends in ASE. It's an enzyme which catalyzes the unwinding or the separation of the hydrogen bonds. It breaks the hydrogen bonds of the parental double helix. Once it breaks it, it's then going to add these binding proteins which attach to the sticky ends of the zipper once you unzip it, so that they don't bond back together. Because if they bond back together, you wouldn't get what's called a replication fork, and the whole process of replication would actually shut down. So here's a graphical image of what happens. This black line right here represents the helicase enzyme. It's going to split the DNA molecule in half, and then these binding proteins are going to kind of twist it on its axis and invert it out. So these sticky ends are now out in the world, and then another enzyme is going to come add these red little pieces right here. So that's what's called a replication fork. It's just hundreds of Y-shaped regions where DNA is actually replicated at. Once that's formed, something called DNA polymerase is going to come through. And polymerase is going to add the complementary bases. So if we have an exposed T, it's going to add an A to it to make a double strand. So the polymerase is going to add these red little dots right here. So this might be an A, this might be a C, this might be a G, because it started with a T, a G right here, and a C right there. So once you get that, DNA is then going to be replicated. So you're getting two identical strands from one original copy. So in summarizing the whole process, the DNA double helix is going to begin to unzip. Again, it's done by something called helicase. We know it's an enzyme because it ends in ASE, and helix because it's breaking the double helix down. So the helicase is going to come through right here. It's going to unzip the DNA. By unzipping it, it's going to allow the polymerase to come along and attach the complementary strands. Once the DNA nucleotides are going to be added, then you're going to get two brand new identical copies from one. And just remember, this is DNA polymerase doing all this work. There was an A here, it added a T. There's a C, it's going to add a G right here. It still has a C right here. So now you're going to get two new strands from one original strand. So you're going to get two identical copies from one original strand. So it's going to be something kind of like this. You're going to start with one strand right here. You're going to get those two identical strands. Like so. So leave you with this one question to think about. What would be the complementary DNA strand for the following DNA sequence? Well, if I give you the sequence of GCG, TAT, G, and ask for the complementary strand, how would you do that? Well, we know that A and T always goes together. A goes with T, and C always pairs up with G. So we know we have that. So A to T, C to G. So if we have a G here, we're going to have a C right underneath it. We have a C here, we're going to have a G. So on and so forth until we complete the complementary strand. And as always, if you have questions, please feel free to email me. This has been Coach Heichel. Next time, we will be covering protein synthesis, transcription, and translation.